historical view, and then Zofia will show you applications of this method. So what is genome editing? So definition is a technique to make precise modification in an organism's DNA, or you can say that this is type of genetic engineering in which DNA is inserted, deleted, modified, or replaced in the genome of a living organism. So this is clear. Application. We can apply this technique or technology to study gene functions, which is called functional genomics. Metabolic engineering, it means if we want to improve or change some metabolic pathways in the organism. Crop improvement, of course, and very important, gene therapy. Tools, what we can do if we want to change genes, disrupt the genes or change it. We can use mutagens or endonucleases that induce double-stranded breaks in DNA. So we have two possibilities, and I will describe both of these possibilities and many other methods. So to put all these methods together in a historical order, the first should be mutagenesis. Mutagenesis was introduced in 1927 for the first time. This is just chemical or radiation-induced mutations. The weakness of this technique, this approach, is that you have random mutations everywhere in the genome. Then, in 1987, meganucleases were introduced. It was the first molecular DNA scissors used to replace, eliminate, or modify sequences in a highly targeted way. So it was much better and a bit better improvement of it. Then, in 1996, zinc finger nucleases were introduced to this field. This was first generation programmable nucleases, so we were able for the first time to design actually nucleases that will cut DNA exactly at the same and certain position. And then we can use this cuts, the damage to introduce another fragments or disrupt the gene. Talents in 2010, this is the same approach like here. So talents are programmable nucleases, but in comparison to zinc finger nucleases with improved specificity, but labor intensive and very expensive when you think about money. And finally, CRISPR-Cas9 in 2012 introduced based on the prokaryotic defense system used for editing genomes in vivo. And this is the first system actually ever which is precise, cost-effective, and very efficient. And I think you described this CRISPR-Cas. This is why it's a kind of, my talk should be at the beginning actually of the course, but okay, but we will repeat something, but I think I will describe other techniques as well. So CRISPR-Cas is so powerful, so wonderful technique that the 2020 Nobel Prize in Chemistry has been awarded to Emmanuel Charpentier and Jennifer Doudna for the development of the method for genome editing for the CRISPR-Cas9. And so this is the best way or the best example that to show how important it was for the science. It was, I would say, even one of the biggest discovery actually in this exactly century, probably. So first, let's concentrate on mutagenesis. As I said, this is chemical or radiation-induced mutation. First person the types of mutagens. So in mutagenesis, you have to use mutagens, and then mutagens are physical, chemical, or biological. The first person to use this mutagenesis is Hermann Josef Müller was the first to artificially induce mutation in flies using X-ray. And it was in 1927, so this was 100 years. That's amazing, you know, that he did something like this kind of experiment. You have several possibilities to use radiation, or several mutagens, physical mutagens to use. Ionizing radiation like alpha rays or beta rays, and no particular, I mean, and gamma rays, X-rays, of course, fast neutrons as well. So this is ionizing radiation, or non-ionizing radiation, which is UV. UV will introduce some mutations as well. The difference is that if you use UV radiation will cause the formation of pyrimidine dimers, leading to point mutation or small insertion or deletions. If you use ionizing radiation like X-rays or gamma rays, you will induce double-strand breaks, resulting in larger chromosomal rearrangement or deletions. So depends what you want to obtain. You should use UV light, or you can use the ionizing radiation, I mean the X-rays or gamma rays or beta or alpha, any of it. What I should say here actually that radiation makes changes directly by the rays and indirectly because it induces some ROS. It means reactive oxygen species, and then they can induce some changes in bases because of it. So we should remember that you have these breaks in the case of radiation, particularly in X-rays or gamma rays radiation, and in addition you have some damage of bases because of reactive oxygen species production. Maybe I should explain pyrimidine dimers. Do you know what it is? When you use the UV and you have two T's, for example, in DNA, then you have the covalent bonds between them, and then during replication, it's a mistake in polymerase, DNA polymerase will make a mistake in this point because it cannot decide whether put A or no A or two A's or something, and then you have deletions or insertions in this place. Okay, so if you use UV, you have a small insertion or deletion. If you use this ionizing radiation, you will induce double strand and huge, even fragments of chromosome can change positions from one chromosome onto another. So it's a translocation, exchange of fragments of chromosome between them, and so on. So it can be lethal, actually. If the dose is very high, it will be lethal. Advantages of physical mutagenesis. Can produce a broad spectrum of mutations, so it's a random, which is kind of advantage because then we can select mutants we really like or we are interested in. And it is suitable for creating large genetic changes. So, but we cannot control it at all, actually. Disadvantages, no specific, as we said, requires labor-intensive screening. It means if you want to obtain a particular mutant, it's a lot of work to do it because you have a random mutations, you have a lot of mutations everywhere in the genome, and then you have to select the mutation in one gene. It's very difficult. And risk of damaging some cellular components, that's really important. It's kind of dangerous, you know, for the cell, for the organism. Chemical mutagens. The first person to introduce chemical mutagenesis was Charlotte Auerbach, who discovered that mustard gas could cause mutation in fruit flies. Again, 1942. That's really important to see how old actually all these techniques are. Some information about classification of chemical mutagens. Usually what we use more often, and actually right now even in laboratories, alkylating agents like EMS, ethyl methensulfonate, and I will tell you what it does in DNA because it's used even now in lab. We will discuss in which, for what kind of experiments. Base analogs like uridine derivatives, and actually even Zofia told you in one lecture, it was the first lecture to use bromo-U, don't you remember? To see the incorporation of bromo-U during transcription, and it was a kind of identification of newly synthesized transcript. So I will say a little bit about it as well. And some regular chemicals, simple chemicals that will change bases. I will tell you some examples as well. Intercalators, I will not discuss this in details, but you have ethidium bromide and other dyes usually, or chemicals that, do you know what intercalators are? They are, yeah, because in double-stranded DNA between bases, yeah. So it's like ethidium bromide we use to visual to see visual to see to see DNA on our gels. Okay, so first let's say, as I said, alkylating agents, ethyl methyl sulfate, EMS, and why it's so important to understand how it works, because we use it even now in the lab when we want to use the random mutagenesis just to get a lot of different mutants and then to select some mutants from this collection. We use EMS to mutagenize it. How it works? It adds ethyl group to guanine, as you can see here, and this O6-ethyl guanine can base pair not with C, but with T, thymine. So it means that if you introduce by EMS this kind of change, let's say in one position of one strand of DNA, after replication of this molecule, you will have T in the complementary strand. Okay, so after replication. So the T is introduced, then in the next generation you will obtain, instead of GC base pair, AT base pair. So of course this is really nice, but the minus is that we don't know where it is. It will happen in many places in the but for screening is good because it's easier. And you know what EMS we used very often to look for some new proteins involved in some processes. Let's say we have a mutants that will flower, let's say in plant case. We have Arabidopsis mutant that will flower, or that flowers late, okay? Then we are looking for some proteins that are important for this pathway, the flowering pathway. And what we do, we use seeds of this Arabidopsis, we use EMS mutagenesis, and you have obtained on seeds, and then we obtain a lot of different mutants, mutated seeds, and we observed which seeds will give, which seed will give a plant, flowering normally. Okay? So we are screening, like screening the library of these mutated seeds, observing flowering time of next generation, and we will select this one which will show the normal flowering. Or you can imagine any other experiments like this. This is called suppressor screening, and we are looking for suppressor. It means the mutation that will suppress some other defect, okay? And then it's relatively easy when you select this kind of suppressor to find in which gene you have mutation by sequencing, by simple sequencing. Right now, high throughput sequencing is so powerful that we can sequence the mutant and we know everything about it and we can select it. So this is EMS, still it is used in laboratories. Base analogs, as I said, uridine derivatives, and when you use it, 5-bromo-U is the most popular here, derivatives are used, and you have here two possibilities. 5-bromo-U can base pair with adenine, as shown here, so this is the UA base pairing, the regular base pairing, or you can use 5-bromo-U in some enol form. You can, this bromo-U can base pair with guanine. So again, if you introduce 5-bromo-U to DNA, and this is just you add bromo-U to your solution to water your plants or something, then you will have, maybe not so easy, I'm sorry, this is not so easy experiment, but then you will have, sometimes there is no mutation, but in some cases you will, by this base pairing with guanine, you will introduce guanine at the place where T was, okay? So then again, in the next generation, you will guanine and then the GC base pair instead of TA base pair. So again, we introduce another mutation in. And some chemicals. This is a big collection of different chemicals. I will describe HNO2, nitrous acid, and this is simple because nitrous acid causes deamination of cytosine and adenine, and you can see here that and when you have C, then you deaminate, so remove this amine group here, and then you have uracil. When you have uracil, this U can base pair with A, so it's again change from C, G to AT base pair, or adenine after deamination is a hypoxanthine. It's a special kind of base, and it can base pair with cytosine. So you can see that instead of AT, you introduce CG base pair. Again, the disadvantages is that it's everywhere in the genome, and it's difficult to collect it. Advantages of chemical mutagenesis: high mutation frequency, of course, relatively simple to implement, and cheap, because these chemicals are relatively cheap. Disadvantages: mutations are random and may require extensive screening to identify desired changes, unless you use this some screening or looking for some phenotypes. So then it's easier, because then you can select phenotypically, morphologically, you can select some plants or some effects on cell culture or whatever to identify desired changes. biological mutation, I will not spend much time on this subject because in some mutagenesis methods, biological agents can be used. These biological agents, I mean, are transposons or mutator strains of bacteria. So transposon mutagenesis is that mobile genetic elements like transposons can be used to insert randomly into the genome, disrupting gene function randomly. But this technique actually right now is used in bacteria only, I would say. Sophia, I think I'm right. This is just transposon mutagenesis is characteristic for bacteria, actually. Yeah, but so it's not for eukaryotic researchers. Mutator strains, it's some bacterial strains with defective DNA repair systems, generate high mutation rates, so you can use them to obtain some mutants, but again, this is also for prokaryotic organisms only. And I was thinking about whether we have some kind of biological mutagenesis in higher eukaryotes, and actually, yes, I was thinking about tDNA-based mutagenesis of plant genes, and actually you had a lecture about it and a tutorial. Am I right? Yeah. So you understand probably the principles about it, but what I want to say is that we can use Agrobacterium tumefaciens to introduce a fragment of tDNA, because you know that Agrobacterium tumefaciens has Ti plasmid, and the fragment of this plasmid called tDNA is inserted into the genome of plant, infected plant. But, and this is random process. So if you use this kind of infection, Agrobacterium infection, then a lot of, you can obtain a lot of plants with disruption of certain single gene. So then you obtain a collection of plants with one insertion in each gene of Arabidopsis. And right now we have a big collections of Arabidopsis mutants, and each particular mutant has insertion of tDNA in a particular gene, which means that this gene is not active. And you can order this mutant from the collection with no actual little price, actually, that is not very expensive. So in this case, you can use this is biological system to obtain mutant of mutation in one gene. Okay, so that's I think this is, you know everything about it, so I will not say much about tDNA insertion. Meganucleases, first molecular DNA scissors used to replace, eliminate, or modified sequence in a highly targeted way. And just a short, this is a kind of very historical view, so you will never ever use them, never ever. This is just a history. So these meganucleases, but meganucleases occur naturally in mitochondrial chloroplast of some organisms, and they assist in homing, the process in which specific DNA sequences are transferred from one genome location to another. Usually in this homing, the intron sequence is transferred from one gene to another. So let's say you have two copies of gene, one with intron, the same gene, coding the same protein, one with intron and one without it. So homing is a transferring of the intron from this, the sequence from one gene having this intron to another gene without it. So this is homing. And this protein, the meganucleases, were able, or are able to recognize and cut specific DNA sequences, 1440 base pair long. This is a kind of the most popular family. It's not important. The genetic engineering allows the creation of recombinant endonucleases targeted to a chosen locus in the genome. So, of course, the naturally occurred nucleases recognize only specific sequences, but we could change genetically, not genetically, and to do some genetic engineering and then obtain proteins and the nucleases cutting at slightly different sequences. But as I said, bye-bye nucleases. There is just the past. Zinc finger nucleases, discovered in 1996. That was really first generation of programmable nucleases. So this programmable nucleases, it means that we can program these nucleases to find and cut a specific sequence in the genome. So I think I have to say something about zinc finger. What do you know about zinc finger? This is just, yeah.
It was introduced by some other professor in this workshop.
You had? Yeah. Okay. Aha. So just to remind you that zinc finger containing proteins are usually proteins like transcription factors, for example. They can recognize precisely sequence in DNA. And here, and you have usually several zinc finger proteins, fingers, zinc fingers. And actually, this is a kind of specific domain with zinc, coordinated zinc. And you have here the example, the 3 zinc finger, this blue one, yellow and green. These balls are zinc ion, and then actually they can recognize precisely DNA. This is in purple, and you can see here, this is kind of some specific transcriptional factor, and you can see that they can recognize directly specific sequences by interaction with specific amino acid residues. And we can actually even change these interactions by changing the sequence of zinc finger domain. So it was possible. Okay. So zinc finger nucleases I'm talking about consists of 2 key components. The zinc finger DNA binding domain, this is the thing was talking about. So composed of multiple zinc finger motifs, each recognizing a specific 3 base per sequence of DNA. So we can actually, if you have a certain sequence, you can design zinc finger to recognize this sequence. If it's like, let's say, 9 nucleotide long sequence, you will have 3 zinc fingers. If it's longer, you need more zinc fingers, specifically designed to recognize and bind to this sequence. And you will bind these zinc fingers together, 1, 2, 3, 4, 5, as you need. And this fragment will bind to DNA. But then what you need is to cut DNA, and then you need FOC1 nuclease domain, and FOC1 is a bacterial enzyme, restriction enzyme, and usually what the restriction enzyme we used, you remember probably, recognize the sequence that they cut the sequence. But FOC1 is a type 2, I think it's called type 2 restriction enzymes. Yes, it's 2. Thank you. And this can recognize one sequence, but cut somewhere else. And they divided this FOC2 nuclease into 2 parts. So remove the parts of recognition, but use just the part for cutting. And then put together zinc finger binding domain, designed specifically for the sequence, with the FOC nuclease domain. So finally, they obtain fantastic thing. You can see here, maybe this is the picture, will explain easily how it works. You need two nucleases at the same time, different nucleases to cut DNA. One with zinc fingers recognizing this sequence, you can say downstream of the cut, and the second nuclease recognizing this sequence upstream from the cut. Then you have spacer, this is a kind of several amino acids, connected FOC1 activity nuclease domain. And you can see that this FOC1 two domains will interact with each other, forming a dimer. So if one fusion protein like this, fusion protein because this zinc fingers are fused to FOC1 fragment, and here, if these two FOC fragments interact with each other, because of interaction of zinc finger with this sequence and this sequence, FOC1 will introduce double cuts in the, and then when you have a double breaks, then repair enzyme will probably introduce some mistakes in this case. So you have several possibilities. If it's homology directed repair, it will be precise repair if homologous DNA template is provided. It's a very complicated process. I don't have time to describe to you this. But usually non-homologous end join repair will be used, and then it's not so precise, and you will introduce some mutations, deletions, insertion, and so on. So then we can disrupt the gene. Okay, so the system at that time was introduced, was perfect actually. All people were really happy with this, but it was some kind of time consuming to prepare and expensive. And you can see that for each particular locus, you have to prepare new enzymes, and it took a lot of time to prepare this kind of recombinant enzymes. Application, so what people did with this zinc finger nucleases. Gene knockout, gene correction or insertion, transgenic models they produced a lot. Therapeutic application, so early exploration of possibility of using this zinc finger, a little bit crazy because it's a huge enzyme, how to put it in the cell and how to control it. This is, I would say, even crazy idea to use it for this, but again, it was the first tries were done with this in gene therapy. Advantages. High specificity due to the modular zinc finger domain we can design, and targeting capability for sequencing up to 1836 base pairs. So if this targets, I mean the sequence was longer, it means that we could do it more specifically, you know, because longer means that this sequence will be present only at this position and not in others. Limitations. Design complexity. Engineering specific zinc finger domains for each target sequence is time consuming and technically challenging, as I said. Off-target effects. Imperfect specificity can lead to unintended mutations. Cost and accessibility, development and production are expensive compared to newer genome editing tools like CRISPR-Cas as what you use now. Again, bye-bye, no one nowadays will use this kind of technique. But It was a big thing when it was introduced to biotechnology field. Talents, 2010. This is exactly the same idea like with zinc fingers proteins, nucleases, but actually we will use a different binding site. Talents use transcription activator-like effector the name is transcription activator-like effector nucleases. And again, two domains. It's always the same. Binding domain and nuclease domain. Nuclease domain is FOC1 is absolutely the same like I described, and everything is true in this case of talents actually of the talent technology. But instead of zinc finger, we use repeats variable domains from transcription activator-like effectors of plant pathogenic bacteria Xanthomonas should be. Okay, sorry. And you have this binding domain. This DNA binding domain contains short repeats, 33, 35 amino acids long. And actually, specific amino acid pair can bind to single nucleotide. So if you know the code, which amino acids will recognize which nucleotide, or which two amino acids, because it's HD will recognize one nucleotide, and at this position, two other amino acids will recognize another nucleotides. Then you can design, actually using these repeats, design the binding site recognizing exactly the sequence you really want. Okay? So it sounds easy. It was better because each zinc finger recognizes 3 bases, and here was 1 base. So much more precisely, we were able to design the binding site, or the binding domain. And it works exactly the same like in zinc finger. So 2 nucleases are needed to cut at one place. The right talon, it means recognizing the binding, recognizing this sequence, and the FOC1 domain, this sequence, and FOC1 domain, and then here the FOC1 after dimerization will cut DNA, the double strand, and again, this double strand breaks can be repaired by 2 different pathways. One which will repair it, so it means there will be no change, no mutations, and the second with random insertions or deletions if non-homologous end joining pathway is used. By the way, insertions and deletions are called indels together, just to remind. Application, again, this is very similar. Gene knockout and knock-in. What's the difference between knockout and knock-in? Knockout means removing removing expression of this gene, and knockout means remove, knockdown knockdown is to make expression a little bit lower, so it's not 0. Knockout means that expression of the gene is 0. Knockdown means that the expression is lower than wild type, but still you have the expression. And lock-in, introduce a gene to express, to be expressed. Oh, activate, yeah. Okay. So therapeutic application, again, they tried to use it to correct genetic mutation in some disease in models of human cells. Transgenic organisms, of course, functional genomics as usual. But again, this was very fantastic, but it's not used anymore. Advantages. Improved specificity in comparison to zinc finger because of this domain, binding domain composition. Flexible design. These talents can be programmed to target virtually any DNA sequence, in contrast to zinc finger nucleases. Reduce context dependence, so talents were less affected by surrounding DNA sequence than zinc finger nucleases. But limitation, labor-intensive design, the same size and delivery. These enzymes are huge, actually, to deliver to the cell to precisely change something. Particular for gene therapy, this is impossible to use it. High cost, of course, because to prepare it, it's time-consuming and very high cost. And then the CRISPR-Cas, I know the system, and actually it's a big revolution. All these systems before that were tried before are actually dead now, right? All people will use the CRISPR-Cas9, maybe except what I said, EMS mutagenesis. It means chemical mutagenesis to obtain some particular mutants. Then people will use it to get some suppressors, as we said. So I think you know everything about the CRISPR, but that just wanted to just repeat some most important thing. That was discovered by two Japanese guys from Osaka University. And CRISPR means cluster regularly interspaced short palindromic repeats. It can be found in gram-positive, gram-negative bacteria in archaea. So it's prokaryotic system. And as you can probably see this kind of pictures in your previous lectures. The bacteria have CRISPR array, the locus in which they have some repeated fragments marked here in black and some different sequences, spacer sequences marked here in different colors. And then when virus or plasmid appeared in the cell can cut this DNA of virus or plasmid into small pieces. This is done by the Cas complex. This is a nuclease complex transcribed and translated from the Cas operon, and then will cleave this DNA and produce a novel spacer will be introduced here in this CRISPR cluster, CRISPR array. So this Japanese guy discovered that bacteria has this kind of gene with repeated short fragments interrupted by the specific sequences. And they found that these specific sequences are complementary or identical to the sequences of some phages, phage DNA, or some plasmid DNA. That was a big discovery. And unfortunately, they didn't get Nobel Prize. You can see here that the real discoverers didn't get it. But the scientists who introduced the CRISPR-Cas to a eukaryotic field and biotechnology, actually, these two scientists, women, got it. Okay, and so this is the after infection or after conjugation, when the plasmid is inside the cell, the fragment of this DNA introduced in the CRISPR array. Then, I'm talking all the time about bacteria, when new infection, the same phage, or not the same phage, but a phage of the same type, infected bacteria, bacteria using the short fragments complementary to the DNA in fact of phage or plasmid, can target it and cut it into the small pieces. Okay? And this is because of complementary of the RNA as CRISPR RNA fragment to the complementary sequence of the phage or plasmid. The only thing you need to obtain the cleavage is the PAM sequence. You know, this is a short sequence here, which is needed for cleavage of the DNA of inventors. Okay. And then how it happened, how it can happen. Of course, you have the Cas operon, which different Cas nucleases. This Cas1 and Cas2, Cas1 and Cas2 nucleases are important for adaptation. It means that they are involved in the cutting of virus or plasmid DNAs. And Cas9 is important for attacking the inventor for the second time. So I will show you that it's for interference, maturation and interference important. But in addition, you remember to Cas9, the nuclease, what is important is track RNA, is trans-activating RNA. This is a short RNA which will collaborate with Cas9 to attack actually to produce Cas9 RNA complexes and to destroy inventor DNA. Okay, so just to show you how it looks, so this precursors CRRNA, CRISPR RNA, is a long RNA. This is a specific sequences complementary to some phages or plasmids, and you have the same repeated sequences marked here in black. So tracker RNA has complementary sequence to this black repeated sequence, and as you can see here, it can bind here, and Cas9 can bind as well to this complex. But RNase-3 is important for cutting this long fragment into short fragments. And this short fragment containing Cas9, CRISPR RNA, and this tracker RNA can recognize the DNA, targeting DNA, make a complementary sequence because of this RNA is complementary to one strand of this DNA. Again, it can happen only when the PAM sequence is present in the DNA, attacked DNA. Then you have this famous R-loop formation. It means that RNA is here, DNA is here, and this strand is displaced. And then you have the Cas9 can cut both strands of DNA here and here. This is one enzyme can cut in because it has two domains, endonuclease domains, and then destroy the DNA. Okay, so that probably this you can know all of this. Maybe I should add, I forgot to say that this RNase-3 not only cut here the precursors of CRISPR RNA, but also it's involved in maturation of tracker. So you can see here that the tracker sequence fragment is removed from this. It's not so important for biotechnology, but to understand the picture. And here you have how we use it for biotechnology. So in natural situation in bacteria, you have CRISPR RNA and tracker RNA in two different molecules. But in biotechnology, we used one molecule. So it's CRISPR and tracker RNA is linked together artificially. And this can be used because we can design the sequence of this complementary to the target, and then we can actually close this complex. We can introducing the complex close to DNA and cut DNA at the certain position, and any position actually whenever we have. The problem is only the PAM sequence, as I said. This is a short sequence which must be present around the cuts. And then just to summarize the possibility of using CRISPR-Cas, you probably, I attended actually the lecture. Someone showed you actually in details vectors and techniques, but I just wanted to summarize everything in one picture. So this is what we can do. We can use Cas9 and so-called guide RNA, designed to specifically bind, make complementary base pairing with target, and Cas9 to cleave DNA in certain position. Again, the problem, we can cut DNA at position only when we have a PAM sequence around, but this is very easy because it's very short sequence, not a problem at all. So then we can, in the cell, when we do it, this double strand breaks will be repaired by non-homologous join recombination, NHEJ. It means that the fragment will get insertion or deletion. This is what we need actually and we want. Or what we can do, we can provide actually some other fragments, donor DNA, and we can introduce in this break another sequence. Okay, so we have two possibilities in CRISPR-Cas. It's not only that you can cut it and you will observe mutations, but you can introduce also by homology-directed repair some additional sequence. Of course, to induce this kind of effects and events, usually in the lab, we use two nucleases, cutting only one strand in one position and the second position. So target 1, target A, and target B. And then we can use the donor DNA to improve the introduction of the fragment. And this is what we are doing in the lab actually very often. So we destroy the gene using this technology, or you introduce another fragment using this technology. It means two Cas9 complexes. I should say this Cas9 are mutated, that they cannot cut two strands, but they can cut only single strand, one strand of the duplex. And some other examples, fantastic possibilities. For example, we can use Cas9 that are mutated at both nucleolytic sites. It means they cannot cut DNA, but they can bind at certain position. And we can actually fuse this Cas9 to activator or inhibitor of transcription. So you can see that what we can do is we can regulate transcription by this complex without destroying DNA, just by putting the complex with activator of inhibitor domain very close to transcriptional sites. And so we can activate or repress. We can even see some specific points on DNA. It means that we fused Cas9 with GFP, for example, or another fluorescent protein, and we will see the hybridization of actually where the Cas9 with the guide RNA is located in the genome. I heard that you discuss the possibility to use Cas9 to stop transcription, so to inhibit transcription by putting a kind of, let's say, transcription is in this direction. So if you can put Cas9 at certain position of the gene, transcription polymerase will stop there. So that's another possibility. I don't have it here. So all these other methods I described are actually dead right now. And you can use only, I think you should use only CRISPR-Cas9 system, which has advantages over all these techniques I described before. Maybe traditional mutagenesis sometimes is used in agriculture, even now, maybe, and in the lab, like EMS mutagenesis. And that's it, I think. So thank you very much for your attention. If you have any questions, I will ask it. I put these colorful things for Holi. Yeah, exactly. For Holi, so happy Holi. This is why I was dreaming about to participate. I was thinking about participating in this Holi event, and this time I'm just at the right time in India. So some questions?